Okay. So, first of all, very good morning to one and all present in
Good
this
one.
session. I, Dr. Rachna Chaturvedi, on behalf of MIT Institute of Biotechnology, MIT University, Uttar Pradesh, and my own behalf, welcome our guest of this session, Dr. Sujit Kumar. Dr. Sujit Kumar is Scientific Officer in Uttar Pradesh Council of Agriculture Research, Lucknow. It's my pride privilege to invite Sir to deliver a talk on the topic CRISPR Cas-9, a potential tool for genome editing. Sir, please.
Thank you, ma'am. I wish to welcome you all in this online presentation, online lecture on the very relevant topic which has given to me by university, that is CRISPR Cas9, a potential tool of genome editing. Ma'am has already introduced me. Myself, Dr. Sujit Kumar, completed my MSc, my PhD in genetics and MBA. And similarly, I have also completed my certificate course in intellectual property rights from Indian Law Institute, Delhi, which is deemed university. And presently, we are dealing with the funding the project, monitoring the projects in the field of genetics, plant breeding, biotechnology, biochemistry, plant physiology. These are all aspects which are related with crop improvement. These are being dealt by me in the UP Council of Agriculture Research, so that I have to become very much aware and upgraded in the field of all sectors of development which are taking place, particularly in the field of crop improvement. Without losing any time, because I wish to give the more the flourishing technology which will go ahead very long after completion the molecular aided technologies for improvement of various living organisms. We are seeing, we are watching the hopefully beyond the this molecular improvement, that is CRISPR-Cas. Even we are going for the base editing as well, which is beyond the CRISPR-Cas. So that I would like to share one or two slides related to base pair as well. Then, as I am telling that we are dealing with the improvement of living organisms slowly and slowly. What are the things? What are the 5Gs we have collected, which are essential for the improvement of any living organism? First one is genome. Genome is the major thing which play major role for the improvement process. If you wish to make any type of improvement that is related with the qualitative or quantitative improvement, means the characters which are being dealt with the qualitative or quantitative inheritance, you must have the holistic idea about the genome of that particular organism which is being targeted for the improvement. Second one, what is Janpad? Mean the variability in the genome. What is the genetic pool, gene pool, which has emerged till today under the forces of the evolutionary forces of the nature? Then the gene pool in the form of germplasm, which is prevailing nowadays, it must to be taken under consideration while you are going for the improvement. Now the genes, what is the total genes which are present? What are the total number of genes which are present in the population, so that you may manipulate those genes? Manipulation may be this influenced by any process, by any induced process, whereas through evolution, the spontaneous, the evolution or the spontaneous manipulation of the gene through nature is a usual process. Now, genomic breeding. This is the very useful term which was the matter of buzz, but presently we have crossed beyond the theory of genomic breeding. Yes, we have to manipulate the genome, the complete genome. That may be the point mutation, through point mutation or frame shift mutation or any other type of genetic manipulation. The genomic breeding is the term of most importance. But nowadays we are targeting for the gene or genome editing. This is the very much useful. Why? Because we are having number of typos, not only in human being. In human being, you are having various typos among 3 billion of the triplets which has been studied till date. But in various organisms, these typos may be eradicated, or some type of point mutation and frame shift mutation may be created with the creation of novel alleles. We are just playing with the alleles which are present in the nature or which has been evaluated through various natural evolutionary forces. But in the process of genome editing, we are able to create new alleles. It means we are able to go beyond the natural selections. Now, promotion of superior alleles, the alleles which are of the dominant type. It means it is hiding the masking the effect of various type of recessive alleles as well, and in the case of in the case of quantitative irritants, these will be acting as the epistatic to hypostat the various alleles present in the more than two alleles, more than two loci on the chromosome, on the genome. Then we can go beyond the heterosis. In the case of heterosis, we are playing with the variability which are present in the nature as well in the female parent or male parent. But in the case of promotion of superior alleles through genome editing, we may create even more superior genotypes. We may create more superior alleles on a particular locus. And the third one is removal of deleterious alleles. Number of deleterious alleles are there. Likewise, in the case of carcinoma, these are the due to activation of this deleterious alleles which are responsible for the shutting down or for the expressing the genes which are related with the onco activities, so that we may create complete knockdown of that type of genes which are sitting for the operation of the oncogene. These all the things may be created with the only help of genome editing. Now, these are some views which can be taken as the importance for the future era, for the future human development. Some the examples which have been this successful with the help of genome editing, which I used to put over here, which are having directly correlation with the human health. 9 boys born with mycopolar myopathy. That is the disease which is related with the weakness of the muscles. Even the person which is having this disease even could not stand with his feet, with the feet of own himself. Then this type of diseases due to only presence of the deleterious genes, which can be completely knocked down, and the disease may be improved. It means this will be very much effective for gene therapy. And China, presently, they have created 86 cancer and HIV patients. The cell library, genetically engineered cell, which use CRISPR-9 technology, it means they silent the gene, which is this inducing the activity of oncogene. But it is under this experimentation only, but it may be created as a hope. It has been successful in plant material, likewise in tomato. Even in the south campus of New Delhi, this type of experiment is going on. And now the one disease which is known as the bubble boy disease, in which the boy is lost its immunity completely. There is no immunity in the fetus from the fetus. Then the boy has to be kept under ventilation, under total aseptic condition to avoid any type of infection, even the normal infection, which is which is bubbling in the environment, with in the air, in the society. So this type of diseases may also be corrected with the help of this genome editing technology. How it is emergence of genome editing as new breeding technology. Now I would like to give some glimpses about the improvement of crops. We will be just discussing about the improvement or correcting the diseases in human being. Now, what is the importance in the crops? Whereas we will also give some slides only to show you that how we are going on with the CRISPR-Cas technology for improvement of crop. That is the editing of the genome, complete genome, whole genome editing is possible for whole genome in the form of insertion, replacement, or removal of DNA for genome with a high degree of specificity. This is the beauty of this technology, that the editing will be of high degree of specificity. Your target gene, your gene of interest will be targeted directly. There is no any chance of off-site targeting of the genes. Now, genome editing by CRISPR-Cas system provide tools relatively low cost. Most of the technology which is being used in the case of genome editing, not only CRISPR-Cas is able to edit the gene. Various technologies are there, like JDFN technology, TALENT technology. These are all related with the editing the gene, but this technology is having this require less technical skills, and similarly it is of the low cost. Now I would like to give some examples of the tools which are being used for this genome editing. The major tools are ZFN means zinc finger nucleases. This work on the basis of meganuclease protein into protein interaction. Since it is based on the protein into protein interaction, therefore heritability may be a problem. And similarly, it is of high cost value. And second, talent based on the protein into mRNA activity, mRNA and protein interaction, but there is involvement of meganucleus. Therefore, all the drawbacks which are related with the ZFN, they will carry on in the case of talent. And the most precise and useful technology present nowadays, these are CRISPR Cas9 technology, CRISPR Cas technology. These are dependent on the protein and DNA interaction. If the DNA is being involved, it means the correction which is being made in the DNA of the organism in the genome will be of heritable type, and similarly it is of low cost. Now, what is the genome editing? I would like to give only glimpses about the genome editing. Genome editing unfold new possibility in agriculture and medicine in both the era, not only in human or animal, but as well as in agriculture. This technology is being flourished and it will be go on. The full form of this is clustered regulatory interspace short palindromic repeats, because the CRISPR gene, which is correlated with this technology, which is the key ingredient of CRISPR-Cas technology, these are the cluster form of the genes. And those genes are, those nucleotides are interspaced and short palindromes are there. The specific palindromes are there, which has to be devised in nature in the case of lower organism, and we have to devise, we may synthesize this in the benefit of human being for genome editing. Now, CRISPR-Cas transfer to system that can be programmed to precisely target, as I've told you that this is very much precise and target specific technology. There is no chance to misfire. If you are having any DNA of any gene of interest, then your complex which has been formulated with integration of CRISPR and Cas9 will directly target on the gene of interest. Now, Cas9 is the CRISPR associated system protein. As I've told you that the CRISPR9 is the component part, and why and how it is the component part, how it will act, I will be discussing in future slide. Then modify gene permanently in living organism. The gene which are present in the living organism, it will itself be modified and heritable. It means we are not going to insert additional gene. It means the natural process which are being operated inside the organism will not be affected, so that there will be very less chance of regulatory mechanism. What are the regulatory mechanism and in which cases these will be required, I will be discussing in the future slides. These are the some technology, comparison of the technology which are based on the indels, insertion and deletions, and these are dependents on the this non-homologous joints, end joints, and homologous repair type. Two type of mechanism may took place in the case of this side-directed nucleases. And if you are having any donor from outside the organism, it means the genetic material which is not present inside the organism, you wish to insert any particular fragment of DNA or fragment of gene, then those type of mechanism may take place over here. In the case of there is no there is absence of donor, so simply the double stranded of DNA may be cut by the this Cas9 enzyme in the nuclease, and after cutting, then if it is being rejoined, then the this homology end regions may delete some one or two, particularly one nucleotide, so that the complete knockdown of the DNA will take of the gene will take place, and the mistakes which are being emerged due to the presence of this mistaken gene or these typos may be eradicated, and the gene therapy may be successful on the basis of this type of therapy. And if you wish to join some part of the DNA on some nucleotides which may act as the component part of that particular gene, which has to be regulated for the in the case in the form of phenotype, then this your side direction nucleases will be gene insertion. And in the case of this homologous repair, the same phenomenon over here, only the same. If you see the B and D, both are same, and this both will be required the regulatory mechanism. And A as C, there is less chance for the regulatory mechanism in the case of homology repairs. Now, what are CRISPR Cas9? I am talking again and again. CRISPR, that is the this RNA type which is present in the naturally, or we can synthesize it, and the Cas9 protein. This complex have to be formulated. For this activation of this complex only, this will only facilitate the gene editing or genome editing, then it is faster, cheaper, and more accurate. As I've told you, this is the site-specific, complete. There's no chance of mistaken. Other techniques having a wide range of potential application. Number of applications are there. We'll be discussing one by one. Then CRISPR Cas9 is a unique technology that enables geneticists and medical research to edit in what are the ways of editing of the genome. The ways of the editing are removal of single nucleotide, or 1 to 9 nucleotide may be removed very easily. 1 to 9 nucleotide may be removed by this process, and adding usually one addition of one nucleotide is enough for converting the activity of the target gene or altering the centenary modification of the gene, so that the phenotypic expression may be modified in the way which we want on the basis of our desire. Then freedom of addition. Hello, 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 hello. It has been disconnected.
Sir.
Hello. Am I audible, ma'am?
Yeah, you are audible.
Oh, but I was not able to listen
Okay.
you. Now this actually for the gene editing previously before CRISPR-Cas, the useful technology was said to be as RNA interference technology, and there are two